Hey y'all, welcome to our presentation on cell respiration by Joanne Leo and Carl Yudmark. Just to briefly recap you students, cell respiration is the controlled release of energy from organic compounds to produce ATP. Now on to Joanne with the next slide.
The first step in cell respiration is oxidation and reduction. Cell respiration involves the oxidation and reduction as they always occur together. Oxidation and reduction involves transfer of electrons from one substance to another. Oxidation is the loss of electrons from a substance and reduction is the gain of electrons. Redox reactions are vital to some of the basic functions of life, for example, photosynthesis and cell respiration, as you can see on the photo, as glucose is being oxidized while oxygen is being reduced. Electron carriers are substances that can accept and give up electrons as required. The main electron carrier in respiration is NAD, which is the abbreviation of
Nicotinamide adenine dinucleotide.
You can see that NAD plus 2 electrons equals reduced NAD, but that is just a brief explanation of what happens. But to give a more in-depth explanation of what happens, initially NAD had one positive charge and exists as NAD+. It gets its 2 electrons from 2 hydrogen atoms that are removed from the substance that is being reduced. One of the hydrogen atoms is split into a proton and an electron, and the other electron, the NAD plus, accepts the electron, and the proton, which is H plus, is released. The NAD accepts both the electron and proton of the other hydrogen atom, which you can see on the other two equations. What this reaction shows us is that reduction can be achieved by accepting atoms of hydrogen because they have an electron. Oxidation can therefore be achieved by losing hydrogen atoms. I'm going to speak about phosphorylation. Phosphorylation of molecule makes them less stable. Phosphorylation is the addition of a phosphate molecule, which is PO4, and it's negatively charged with -3, to an organic molecule. The purpose of phosphorylation is to make the phosphorylated molecule more unstable or more likely to react. The hydrolysis, which is a reaction involving the breaking of a bond in a molecule using water of ATP, releases energy to the environment and is therefore also called an exergonic reaction. The opposite of exergonic is endergonic, which is energy absorbing. The first reaction in the series of reactions is called glycolysis. Glycolysis in eukaryotic cells takes place in the cytosol. Here you can see that glucose is turned into glucose 6-phosphate, and that ATP forms ADP when it breaks a phosphate group to utilize its bond energy to do work. Glycolysis takes help with the help of a family of enzymes called hexokinesis to form glucose 6-phosphate. The conversion of glucose to glucose 6-phosphate is endergonic, and the hydrolysis of ATP is exergonic. The two reactions are coupled and takes place spontaneously. Glycolysis consumes a small yield of ATP but can take place with the use of any oxygen by converting sugar into pyruvate. This is done in many small steps. ATP is used in phosphorylating sugar. First, glucose 6-phosphate is rearranged into glucose 6-phosphate by glucose phosphate isomerase, and that turns into fructose 1.6 biphosphate. The phosphorylation reaction reduced the activation energy required for the next reaction, which makes it more likely to occur. In the next step, the fructose biphosphate is split into 2 molecules of triose phosphate. These 2 molecules of triose phosphate is then oxidized to glycerate 3-phosphate in a reaction that can make enough energy to produce ATP. The oxidation takes place by removing hydrogen. The hydrogen is then accepted by NAD+, which turn into NADH+, and NADH plus H plus, which you can see on the screen. In the final stage of glycolysis, the phosphate group is transferred to ADP to produce more ATP along with pyruvate. The stages take place twice per glucose. In aerobic cell respiration, pyruvate is decarboxylated and oxidized. 2 molecules of pyruvate are produced in glycolysis per molecule of glucose. If oxygen is present, the pyruvate is absorbed into the mitochondrion where it is fully oxidized. Carbon and oxygen are removed. Carbon and oxygen are removed in the form of carbon dioxide, which is on this equation, which is called decarboxylation. Pyruvate is oxidized by removing hydrogen of pairs of hydrogen atoms. The hydrogen carrier NAD plus and FAD accept these hydrogen atoms and pass them onto the electron transport chain where oxidative phosphorylation will take place, which Carl will talk more about later. When the pyruvate has been produced in the cytoplasm, it is brought to the mitochondrial matrix. In the matrix, the pyruvate is decarboxylated and oxidized to form an acetyl group. 2 high-energy electrons are removed from the pyruvate. The 2 electrons then react with NAD plus to produce reduced NAD, which you can see here. So basically, what the link reaction is, pyruvate is converted into acetyl coenzyme A.
Hey there, folks. Carl here. I'm here to talk about the Krebs cycle today. For all of you who are wondering what it is, it's essentially the oxidation of acetyl groups that are coupled to the reduction of hydrogen carriers. Link reactions basically involves 1 decarboxylation and 1 oxidation. But in the Krebs cycle, there are an additional 2 more decarboxylations and 4 more oxidations in the Krebs cycle. Most of the energy released in the oxidations of the link reaction and Krebs cycles is used to reduce hydrogen carriers, which are known here as NAD plus and FAD. As a result, the energy remains in chemical form, so it can be passed to the last part of aerobic cell respiration, or also known as oxidative phosphorylation, which I'll talk more about in the next slide. So to put this into basic context, it's for every turn of the cycle, the production of reduced NAD occurs 3 times, decarboxylation occurs 2 times, and reduction of FAD occurs 1 time, plus 1 ATP is produced. On to the next slide of oxidative phosphorylation. Energy released by oxidation reactions are carried to the cristae, which is basically the partial partitions in a mitochondrion, by reduced NAD and FAD. Reduced NAD is produced in glycolysis, link reactions, and Krebs cycle, while the FADH2+ is reduced only in the Krebs cycles. As stated before, this is the final part of aerobic respiration. This is because ADP is phosphorylated to produce ATP, using energy from oxidation. The substances oxidized include FADH2+ and reduced NAD. These molecules are used to carry energy released in these stages to the cristae. Now onto the electron transport chain. The energy from the oxidative phosphorylation is released in a series of small steps done by a chain of electron carriers. Reduced NAD and FADH2 give electrons to the electron carriers. As electrons are passed to each carrier, energy is maximized to transfer protons across the inner membrane from matrix to inner membrane space. Protons flow through ATP synthase down concentration gradient, providing energy needed to make ATP. Now on to the last slide of chemiosmosis. This happens because of a chemical compound, which in this situation is H+, moves across the membrane down the concentration gradient. This releases the energy needed for ATP synthase, the enzyme, to make ATP. As you can see in the diagram, NADH plus H+ supplies pairs of hydrogen atoms to the first carrier NAD+, and it is then returned to the matrix. Hydrogen is then split to release 2 electrons, which is passed from carrier to carrier. As electron is transferred, energy is released. 3 of these use the energy to transfer H plus across inner mitochondrial membrane from the matrix to the inner membrane space. As the electrons flow with more protons pumped across the membrane, concentration gradient of protons build. It becomes a share of potential energy. In order for the electrons to flow, they must be transferred to terminal electron acceptor at the end of the chain. This is the O2, which briefly becomes O minus 2, but combines with 2H plus to make water. Protons pass back from the intermembrane to matrix by ATP synthase as they move down the concentration gradient. Energy is released and is used by ATP synthase to phosphorylate ATP.